Settle in for a moment. Let your shoulders drop just a little. Let your breathing slow to something unhurried. Let the quiet around you deepen until it becomes something thick and comfortable, like a blanket pulled close on a cool night. Tonight, we are going to travel somewhere vast, not across a map, not from one country to another, but across time itself. We are going to travel backward across hundreds of thousands of years, to a world so different from the one you know that it would be nearly unrecognizable to any modern eye. No cities, no roads, no written history, no agriculture, no metal tools. Just open land, shifting seasons, vast stretches of wilderness extending from horizon to horizon, and the slow, patient turning of generations of human lives, each one short and hard and full of effort. Each one contributing almost imperceptibly to the shape of the world that would eventually come. And somewhere in all of that enormous passage of time, something happened. Something quiet and invisible. Something that did not announce itself and that nobody witnessed as a singular event, because it was never a singular event. It was a process. A long, slow, unimaginably patient biological process that unfolded not in a single dramatic moment, but across thousands upon thousands of years, in bodies that never knew it was happening, in lives lived simply in the effort to survive, to find food, to find shelter, to protect children, to outlast the cold and the heat and the disease and the predators that shared their world. Somewhere in that immense stretch of time, human skin changed, not just a little, not in one place. Across nearly every corner of the earth that humans eventually reached, in populations separated by oceans and mountain ranges and tens of thousands of miles of impossible distance, skin color shifted, diversified, and adapted in ways that still shape how people look today. The extraordinary palette of human skin tones visible across the living human species, from the deepest browns and blacks of equatorial Africa to the palest pinks and creams of northern Europe and everything possible in between, did not always exist. It is not an ancient given. It was made, slowly, by forces that operated entirely below the level of individual awareness, in the arithmetic of survival and reproduction across generation after generation after generation. The story of how humans came to have the range of skin tones they carry now is one of the most thoroughly researched questions in all of evolutionary biology. It touches on genetics and ancient geography, on nutrition and reproductive health, on the physics of solar radiation and the chemistry of vitamins that we still depend on today, on the massive population movements of deep prehistory and the way those movements reshaped the genetic landscapes of entire continents. It is a story that has been painstakingly assembled from bone fragments and extracted DNA, from satellite measurements of ultraviolet radiation, and from the genomes of people living today, from archaeological sites and cave paintings and ancient agricultural settlements built by people who had no idea history was being made. Every piece of the picture has been hard-won, the result of decades of patient scientific work across disciplines that rarely communicated with one another until recently. And that picture, now that enough of it has come into focus, turns out to be nothing like what most people would guess. Here is the finding that tends to stop people most completely when they first hear it properly explained. Light skin in Europe is genuinely, profoundly recent, not ancient, not gradual from the moment early humans first arrived on that continent, not a slow and steady lightening that began when people first moved away from equatorial Africa and progressed in smooth increments over the following tens of thousands of years. What the ancient DNA evidence now tells us, with increasing clarity and from a growing number of studied samples and sites across the continent, is that pale skin did not become widespread in Europe until roughly 3,000 years ago. 3,000 years ago. That is approximately 1000 BCE. That is within what historians call the Iron Age. That is a time when the Phoenicians were actively sailing the Mediterranean, when the Zhou dynasty was ruling ancient China, when the earliest Greek city-states were in their formative period. That is, on the scale of human evolutionary history, essentially the day before yesterday. And for the roughly 42,000 years before that, during which modern humans had been inhabiting Europe. For most of that vast stretch of time, the evidence increasingly suggests The majority of Europeans had dark skin. A comprehensive genetic study published in 2025, examining 348 ancient human genomes from individuals who lived between roughly 45,000 and 1,700 years ago. Drawn from archaeological sites across 34 countries in Western Eurasia, found that for much of the period covered by the study, individuals with dark skin represented the clear majority. Approximately 63% of the ancient people studied had dark skin, while only about 8% had genuinely pale skin. As recently as 5,000 years ago, during the Copper Age, about half of the sampled European population still carried genetic profiles indicating dark or intermediate skin tones. The famous glacier mummy, known as Ötzi, perhaps the single most extensively studied ancient European individual in all of history, died approximately 5,300 years ago in the Alps. His body was preserved in ice for millennia and recovered in 1991 in a remarkable state of preservation. He has been examined in extraordinary detail. And when researchers analysed his genome fully in 2023, they found that his skin pigmentation genetics indicated a tone darker than that of modern Southern Europeans. One researcher described it as the darkest skin tone recorded in any contemporary European individual whose genome has been genetically analysed. Ötzi, the famous Iceman, a man sophisticated enough to carry copper tools and woven grass clothing and various herbal medicines, would have had skin considerably darker than most modern Italians or Greeks. Or consider the finding that became globally famous in February 2018, when researchers at University College London and the Natural History Museum extracted and sequenced the genome of a man who died roughly 10,000 years ago in what is now Cheddar Gorge in Somerset, England. This individual, known as Cheddar Man, had been assumed for over a century to have been fair-skinned, because he was European and because light skin had been imagined as a fundamental feature of European populations since ancient times. The DNA told a completely different story. Cheddar Man, living and dying in what would eventually become England, had dark to dark black skin, dark curly hair, and blue or blue-green eyes. His genetic profile matched that of a wider group of people known as the Western hunter-gatherers, who ranged across Western Europe during the Mesolithic period and who consistently show the same pattern across multiple ancient DNA studies. Dark skin, frequently combined with light eye color. The combination of dark skin and blue eyes seems paradoxical to anyone accustomed to the way these traits cluster in modern European populations. But it was, apparently, not unusual at all in Mesolithic Europe. It was a recognizable, even characteristic appearance among the Western European hunter-gatherers of that period. All of this raises a fundamental question that deserves a thorough answer. How did we get from there? From a Europe populated by dark-skinned hunters and gatherers who, in terms of their basic appearance, would have looked entirely at home in parts of East Africa or South Asia, to the pale-skinned, light-haired, light-eyed modern Europeans who are now so strongly associated with that same continent. And beneath that question lies a deeper one that the surface question depends on. Where did human skin color come from in the first place? What forces shaped it into existence? What biological logic does it follow? And why does it vary across the planet in the specific geographic patterns that it does? Darker near the equator, lighter as you move toward the poles, in a gradient so consistent that you can predict the average skin tone of a population from its latitude, almost as reliably as you can predict its average temperature? Those are the questions we are going to move through tonight, slowly, carefully, with all the patience that a story measured in hundreds of thousands of years genuinely deserves. Section 2. The original blueprint. Life under the African sun. To understand where human skin color comes from, you have to go back much further than the Mesolithic period, much further even than the emergence of modern humans, to a moment in deep evolutionary history when something changed in our ancestors that set in motion everything that eventually followed. The genus Homo, the genus that includes modern humans and all of our extinct evolutionary relatives, is thought to have originated in Africa somewhere around 2.8 million years ago. The earliest members of this genus were different from us in many respects. Smaller-brained, differently proportioned, toolmakers only in the most basic sense. But they were distinctly on the path that would eventually lead to us. And they had something we do not have, or at least something most of us have only in vestigial amounts: body hair. Our primate relatives, the chimpanzees and bonobos and gorillas, have hair covering most of the surfaces of their bodies. Our earliest ancestors almost certainly did too. This is the ancestral condition of primates and of mammals generally. Hair is the default. The thing about a chimpanzee, if you were to shave it, is that the skin underneath is relatively pale. Not dramatically so in all cases, but considerably lighter than the skin of a modern equatorial African human. And this makes biological sense. Hair provides significant protection against ultraviolet radiation, scattering and absorbing UV before it reaches the skin surface. If the skin is covered by dense hair, the skin does not need to provide UV protection itself. The hair is doing that work, and so the skin beneath can be relatively lightly pigmented without that being a problem. This was the likely starting point of human pigmentation. Pale skin covered by dark hair, similar to what you would find in our closest primate relatives. The evolutionary challenge that changed this was the loss of that body hair. Somewhere over a period estimated to stretch from approximately 4 million to 1.2 million years ago, the human ancestral lineage lost most of its body hair. The precise timeline is very difficult to establish because soft tissue does not survive fossilization. Hair does not leave bones. It does not leave impressions. It essentially vanishes, leaving no direct record. Scientists have to approach this question indirectly, using genetics to estimate when certain hair-related genes changed, using physiology to reason about when the distinctive human eccrine sweat gland system expanded, and using comparative biology to understand how these changes fit into the broader picture of hominin evolution. The best current thinking ties the loss of body hair to a shift in lifestyle that was happening in the human lineage during this period. As our ancestors spent more time in open savanna environments and less time in the shade of forests, as they became increasingly capable of extended travel and what researchers call persistence hunting, running prey animals down over long distances in the heat of the day. Rather than ambushing them, they faced an acute thermal management problem. The human metabolic system, particularly during vigorous physical activity, generates enormous amounts of heat. Too much internal body heat is dangerous. Brain tissue is particularly sensitive to temperature. Even modest overheating can cause cognitive impairment, and severe overheating can be rapidly fatal. The solution that evolution found was the expansion of an extraordinary system of eccrine sweat glands. These are the glands that produce the thin, watery cooling sweat that covers your skin when you exercise intensely in the heat. Most mammals have relatively limited numbers of eccrine glands. Humans have somewhere between 2 and 4 million of them, distributed across the body surface with a density that is remarkable among mammals. When activated, this system can move enormous amounts of water to the skin surface, where it evaporates and carries heat away from the body with tremendous efficiency. But for evaporative cooling to work well, the skin surface needs to be exposed. Hair is an insulator as well as a covering. It traps heat. It creates a microenvironment of warm, humid air between its fibers and the skin. It reduces the efficiency of sweat evaporation. As our ancestors became more dependent on active sweating for temperature regulation during extended physical exertion, dense body hair became a liability. Over generations, individuals with less body hair could run longer, cool more efficiently, overheat less readily. The selective pressure against dense body hair accumulated until, gradually, the lineage became essentially hairless across most of the body surface. And this created a new problem. A serious one. Suddenly, a large expanse of skin that had previously been covered and protected by a coat of dark hair was exposed to the full intensity of the equatorial sun. And the equatorial African sun is intense. At latitudes near the equator, ultraviolet radiation falls on the Earth's surface with remarkable power, nearly year-round, in quantities that are orders of magnitude greater than what reaches the surface at higher latitudes. Ultraviolet radiation does several things to exposed skin. It damages DNA in skin cells, potentially initiating the molecular cascade that leads to skin cancer over time. It breaks down collagen and other structural proteins. It causes the visible damage we associate with excessive sun exposure. But the effect most immediately relevant to evolutionary fitness in our ancestral environment is something more specific and more directly connected to reproduction. It destroys folate. Folate, also called folic acid or vitamin B9, is essential to life in multiple ways. It is critical for DNA synthesis and repair. It is essential for cell division. It is necessary for the production of healthy, motile sperm. It is required in particularly large amounts during early fetal development, when cells are dividing rapidly to form the embryo, and its absence at that stage dramatically increases the rate of catastrophic neural tube defects, including spina bifida and anencephaly, conditions that destroy or severely impair the developing fetal nervous system. And folate is not stored safely in some deep internal reservoir. It circulates in the blood, including in the subcutaneous blood vessels that run just beneath the skin surface, close enough to the surface that UV radiation penetrating the skin can reach them. Two researchers named Richard Brander and John Eaton, publishing in 1978, were the first to formally document what UV radiation does to folate. And they showed that light-skinned people exposed to simulated intense sunlight had abnormally low levels of folate in their blood. More strikingly, they showed that exposing human blood serum directly to the same simulated UV conditions caused a 50% reduction in folate content within just 1 hour. Not overnight, not over days. 1 hour. The evolutionary logic of what follows from this is direct and compelling. Under intense equatorial UV radiation, individuals with pale, unprotected skin would consistently have their folate degraded. Lower folate in women means higher rates of pregnancy loss and fetal malformation. Lower folate in men directly impairs sperm production and motility. Either way, individuals with inadequate folate protection leave fewer healthy surviving offspring. Their genes, and including whatever variants led to their inadequate folate protection, become progressively less represented in successive generations. Over time, the population shifts toward better protection. The solution was already available. Melanin, particularly the dark form called eumelanin, is one of the most efficient UV-absorbing materials in biology. Eumelanin can dissipate over 99.9% of absorbed ultraviolet radiation as heat before it can reach the blood vessels just beneath the skin surface. More eumelanin in the skin means more folate protection. More folate protection means better reproductive success. Better reproductive success means the genes for more eumelanin production spread through the population. The genetic evidence for this process is remarkably consistent. Analysis of the melanocortin-1 receptor gene, a central regulator of melanin production, suggests that a strong selective sweep toward dark, eumelanin-rich skin occurred in the human lineage approximately 1.2 million years ago. This timing aligns closely with other estimated dates for the major loss of body hair and the expansion of the eccrine sweat gland system. All of these changes appear to have converged on a similar period, painting a consistent picture. An increasingly active, increasingly hairless hominin living in open equatorial Africa, whose survival increasingly depended on staying cool and whose reproductive success increasingly depended on protecting folate from the sun. By the time anatomically modern Homo sapiens emerged, approximately 300,000 years ago, dark skin was the ancestral and universal condition of the species, not one of several options. Not a variation among variants. The default, the starting point. Every person alive today, regardless of what their skin looks like, is descended from those dark-skinned African ancestors. The diversity of human skin tones that exists across the living human species is not something that was present from the beginning and then maintained in different populations. It is something that arose later, as populations spread across the globe and encountered radically different environments, and as their bodies slowly, over countless generations, adjusted to the UV conditions of those new environments. The changes came after the migration, not before it, and in most cases they came much later than anyone had previously assumed. Section 3. The biology of skin color. The machinery of pigment. Somewhere in the deepest layer of the outer skin, a thin sheet of specialized cells called melanocytes maintain a quiet but essential function. They are not the most abundant cells in the skin. They make up perhaps 5 to 10% of the cells in the basal layer. But they hold an outsized responsibility for one of the most fundamental protective systems the body possesses. Melanocytes exist to make melanin and distribute it. That is their biological purpose. And here is something that genuinely surprises most people when they encounter it for the first time. The number of melanocytes in a given area of skin does not differ significantly between people with very different skin tones. A person with the deepest brown-black skin and a person with the palest possible pink-white skin carry roughly comparable numbers of melanocytes in any given square centimeter of their bodies. What differs between them is not how many of these cells they have. What differs is what those cells are doing, how active they are, how much melanin they produce, what type of melanin they make, and how efficiently they distribute that melanin to the surrounding skin cells. The production of melanin begins with a simple amino acid called tyrosine. Tyrosine is one of the standard 20 amino acids that make up proteins. It is present in virtually any protein-containing food. Inside the melanocyte, tyrosine is taken up and subjected to a series of enzymatic reactions. The critical first steps are catalyzed by an enzyme called tyrosinase, which converts tyrosine into dopa and then dopa into dopaquinone. Dopaquinone is the branch point, the moment where the chemistry can go in two fundamentally different directions. Down one path, dopaquinone proceeds through a series of additional reactions to produce eumelanin. This is the dark brown to black pigment. It forms as an irregular polymer, a tangled molecular structure that is not fully characterized even today, but whose function is extremely well understood. Eumelanin is an extraordinarily effective absorber of UV radiation across a broad range of wavelengths. Once it absorbs UV energy, it converts that energy safely into heat through a process so efficient that over 99.9% of absorbed UV radiation is disposed of this way before it can go on to damage other molecules. It is, in biological terms, a near-perfect sunscreen. Down the other path from dopaquinone, when the amino acid cysteine is present in sufficient concentration, the chemistry diverts toward pheomelanin. This pigment is red to yellow in color. It is the pigment responsible for red hair and the characteristic pinkish tones of very fair skin. And it differs from eumelanin in a critical way. Pheomelanin is photounstable. When it absorbs UV radiation, it does not safely disperse that energy as heat. Instead, it can generate reactive oxygen species, molecular fragments that are chemically reactive and that can attack and damage DNA and other biological molecules. This is one of the primary reasons that people with high pheomelanin content, particularly those with red hair and very pale skin, have dramatically elevated rates of melanoma, the most dangerous form of skin cancer. The ratio of eumelanin to pheomelanin produced by a person's melanocytes, which is controlled primarily by genetics through the activity of a receptor protein called MC1R and other signaling molecules. OS determines much of their apparent skin color. Dark skin has abundant eumelanin and relatively little pheomelanin. Pale skin has less eumelanin and relatively more pheomelanin in the overall melanin composition. The gradation between these extremes represents the full range of human skin color. After melanin is produced, it is packaged into small membrane-bound organelles called melanosomes. These are the delivery vehicles. Melanosomes are then transported along the long branching projections that extend from each melanocyte outward into the neighboring tissue, and they are transferred from the melanocyte into the keratinocytes, the cells that make up the main body of the outer skin layer. Once inside the keratinocytes, melanosomes migrate to a specific location above the cell's nucleus. It's a clustering there in what researchers call the supranuclear cap. This is not a random arrangement. It is a deliberate protective structure. The melanin in this cap is positioned precisely to intercept UV radiation arriving from above before that radiation can penetrate to the nucleus and damage the DNA stored there. In people with deeply pigmented skin, this system operates at full capacity. The melanosomes are large, numerous, densely packed with eumelanin, and resistant to the lysosomal enzymes that would otherwise break them down quickly. They persist intact throughout the lifetime of the keratinocyte, providing consistent protection from the outermost skin cells down through the deeper layers. Measurements show that melanin in deeply pigmented skin allows only about 7.4% of ultraviolet B radiation and roughly 17.5% of ultraviolet A radiation to pass through the skin surface. In pale skin, those numbers jump to approximately 24% and 55% respectively. The protective difference is not subtle, it is substantial. The epidemiological consequences of this difference are measurable and significant. Studies consistently find rates of melanoma and other skin cancers that are 20 to 60 times higher in pale-skinned populations compared to deeply pigmented populations living in similar UV environments. Melanoma, the most lethal form of skin cancer, kills tens of thousands of people per year in modern developed countries, and the vast majority of those deaths occur in pale-skinned people. The protection that dark skin provides against this outcome is real and substantial. Understanding this makes the original logic of dark skin perfectly clear. For ancestral human populations living under the intense UV radiation of equatorial Africa, dark skin was protective in multiple ways simultaneously. It shielded folate from photodegradation. It protected DNA in skin cells from UV damage. It reduced the risk of eventually fatal skin cancer, though cancer risk is less relevant evolutionarily since skin cancers typically arise after reproductive age. The folate protection was the evolutionarily dominant benefit, directly affecting fertility and fetal development across the whole population in every generation, which makes the question that follows all the more interesting. If dark skin is such a clear biological advantage, what could possibly be strong enough to drive evolution away from it? What environmental pressure could outweigh more than 1,000,000 years of selection for dark, folate-protecting skin? Section 4. The Great Balancing Act. Folate, vitamin D, and the geography of light. The answer begins with something that is easy to forget in a world of supplemental vitamins, fortified foods, and year-round nutritional abundance. In the ancestral world, there was one nutrient that could not reliably be obtained from food alone and that could not be substituted, synthesized, or manufactured by any cultural technology. It had to come from the sun. That nutrient is vitamin D. Vitamin D is technically classified as a prohormone rather than a true vitamin. That is because, unlike most vitamins, it can be synthesized within the body rather than having to be obtained from the diet. But the synthesis depends entirely on one specific trigger: ultraviolet B radiation from the sun striking the surface of the skin. When UVB radiation hits the skin, it converts a cholesterol-derived molecule called 7-dehydrocholesterol into provitamin D3. This molecule then undergoes a thermal isomerization reaction driven simply by body heat, converting into vitamin D3. From there, vitamin D3 travels in the bloodstream to the liver, where it undergoes a first modification, and then to the kidneys, where it undergoes a second modification that produces the biologically active hormonal form. The entire pathway is elegant in its biological efficiency. Bare skin plus appropriate sunlight equals vitamin D. Why does vitamin D matter so much? The list of its functions is long and has grown substantially as research has continued. Most fundamentally, it regulates calcium absorption in the gut, which means it is essential for building and maintaining strong bones. Without adequate vitamin D, calcium cannot be properly absorbed regardless of how much calcium is consumed, and the result is demineralized, weakened bones. In children, severe deficiency leads to rickets, a condition of profound bone softening and deformity that would have been crippling and often fatal in an ancestral world that required vigorous physical activity for survival. Adults with chronic vitamin D deficiency develop osteoporosis, weakened immune function, elevated susceptibility to respiratory infections, including tuberculosis, and increased risk of multiple forms of cancer. Research published in recent decades has revealed vitamin D receptors in almost every tissue in the body, and estimates suggest that approximately 5% of the human genome is under the influence of vitamin D signaling. It is not a peripheral nutrient. It is deeply central to human physiology. Now here is the tension that matters for our story. Melanin, the very molecule that protects folate by absorbing UV radiation before it reaches the subcutaneous blood vessels, also absorbs the UV radiation that would drive vitamin D synthesis in the skin. Dark skin is a superior shield against UV across all wavelengths. But that means dark skin also blocks more of the UV that could otherwise be converted into vitamin D. At equatorial latitudes, under 12 months of intense tropical sun, this trade-off does not create a problem. UV radiation is intense enough, delivered in large enough quantities year-round, that even deeply dark skin allows sufficient UV penetration to support adequate vitamin D synthesis. The primary selective challenge at these latitudes is too much UV, and dark skin is clearly favoured as protection against it. But move away from the equator. Move north or south into temperate latitudes. Move to places where the sun sits lower in the sky, where the sun's rays must travel through a longer path of atmosphere before reaching the ground and lose much of their UV intensity along the way, where winter brings months of short days and weak sun and cold that keeps people bundled in clothing rather than exposing skin to whatever weak sunlight exists. In these environments, the situation is fundamentally reversed. UV radiation is scarce. Getting enough of it through the melanin barrier of dark skin to drive meaningful vitamin D synthesis may be impossible for significant portions of the year. A dark-skinned person in Northern Europe in winter months may, in the absence of dietary compensation, be essentially unable to synthesize vitamin D through skin exposure at all. And chronic vitamin D deficiency in this situation leads directly to rickets in children, to impaired immune function, to higher mortality from infections, and to reduced reproductive success in multiple ways. Natural selection, acting on these outcomes, will consistently favour whatever genetic change results in lighter skin and better UV penetration for vitamin D synthesis. This is the central insight of the vitamin D folate hypothesis, first formally articulated by researchers Nina Jablonski and George Chaplin at Penn State University in 2000, and refined through 25 years of subsequent research. The hypothesis proposes that the entire global pattern of human skin color diversity is essentially the expression of natural selection balancing two competing metabolic imperatives. Maintain enough melanin to protect folate from UV degradation in high UV environments, and maintain little enough melanin to allow UV penetration for vitamin D synthesis in low UV environments. Near the equator, where UV is intense, the folate protection pressure dominates. Dark skin is favoured. At high latitudes, where UV is weak, the vitamin D synthesis pressure dominates. Lighter skin is favoured. In between, the intermediate tones represent the calibration point for intermediate UV environments. To test whether this was actually what was happening, Jablonski and Chaplin obtained NASA satellite data providing precise measurements of ultraviolet radiation reaching the Earth's surface across the entire globe, across every latitude and longitude, across all seasons. They then compared these UV measurements against comprehensive data on the skin color of indigenous populations at those locations around the world, drawing on reflectance measurements that quantify skin darkness in a standardized way across different populations. The correlation they found was extraordinary. The relationship between UV radiation levels and indigenous skin color was among the strongest correlations in all of evolutionary biology. Darker populations at higher UV latitudes, and lighter populations at lower UV latitudes. This pattern held across Africa, across Asia, across the Americas, across the Pacific, across virtually every region studied. The exceptions were predictable and themselves informative. Populations that had migrated relatively recently to a new UV environment had skin calibrated for their origin, not their current location, exactly as the hypothesis would predict. One particularly revealing detail in the analysis was that skin color correlated more strongly with autumn UV levels than with annual average or summer peak UV. This makes intuitive sense when you think about it carefully. The critical constraint for high-latitude populations is not summer, when UV is highest and even dark skin can produce some vitamin D. The critical constraint is winter, when UV is weakest and vitamin D stores must carry the population through months of negligible synthesis. What determines how much vitamin D is stored going into winter is primarily the autumn UV levels, when the body is still actively synthesizing and storing vitamin D before the season closes. A population at a latitude where autumn UV drops below the synthesis threshold early and stays there will be consistently vitamin D depleted through winter. Selection for lighter skin that allows more of the limited autumn UV to penetrate would be particularly intense at those latitudes. The reproductive biology makes the selective pressure concrete. Vitamin D deficiency in pregnant women has been linked to pelvic malformations that can complicate delivery. It impairs immune function in ways that increase mortality from diseases that strike hardest during the reproductive years. Folate deficiency, which dark skin helps prevent, causes devastating neural tube defects in fetuses and impairs male fertility. Both vitamins, both UV-dependent, both directly affecting how many healthy offspring a person produces. Natural selection acting ruthlessly on those outcomes would consistently favor the skin tone that optimizes the balance between them for whatever UV environment a population inhabited. The vitamin D folate hypothesis does not claim to explain every detail of human skin color variation. Sexual selection, random genetic drift in small populations, and specific dietary histories also contribute to the variation we see. But it provides the most coherent, most parsimonious, and best supported explanation for the major geographic patterns of human skin color that has yet been proposed. And it generates testable predictions that the data, accumulated over decades of research by many independent groups, consistently support. With this foundation in place, we can now follow the actual story of how populations moved out of Africa, how their skin color responded to new environments, and how the specific events of human prehistory shaped the pigmentation patterns we see in the world today. Section 5. Out of Africa, the long walk into a changing world. By approximately 300,000 years ago, anatomically modern Homo sapiens had emerged in Africa. The precise location within Africa of this origin is still debated, with some evidence pointing to East Africa and other evidence suggesting it may have been multiple locations across the continent. But the broad picture is clear. Our species began in Africa, and for hundreds of thousands of years after its emergence, it remained there. The rest of the world was not uninhabited. Neanderthals were well established in Europe and Western Asia, having adapted over hundreds of thousands of years to the cold climates and low UV conditions of those latitudes. Denisovans occupied Eastern Asia and Siberia. Possibly other human relatives existed in other regions. But anatomically modern Homo sapiens, the lineage that includes every living person, began in Africa and remained an exclusively African species for a very long time. Then, beginning at some point between perhaps 120,000 and 60,000 years ago, the edges of that exclusivity began to erode. There appear to have been early excursions from Africa into the Arabian Peninsula and the Levant, some evidence of which has been found in the form of Homo sapiens fossils and associated artifacts in sites like Mislia Cave in Israel and various Arabian sites. But these early forays appear to have left no lasting genetic contribution to populations outside Africa today. They may have gone extinct, been reabsorbed, or simply retreated back as conditions changed. The movement that matters decisively for the world we live in, the one that produced all non-African human populations today, happened somewhere around 60,000 to 70,000 years ago. A relatively small group of modern humans, perhaps numbering only in the hundreds or low thousands at the bottleneck moment of the crossing, left Africa. The most favored route by current evidence leads through what is now the Horn of Africa, or across the southern Strait of the Red Sea, and then along the coastlines of the Arabian Peninsula and South Asia, following the shorelines of the Indian Ocean where food in the form of coastal and marine resources was abundant. These people were dark-skinned. This requires no inference or assumption beyond understanding the ancestral state of the species as we have explored it. After over 1 million years of strong selection for dark, eumelanin-rich skin under the equatorial African sun, the founding population of the out-of-Africa migration carried dark pigmentation. They were leaving the place that had made them dark. They had not yet encountered the selective pressures that would eventually drive change. The migration quickly diversified into multiple directions. One branch, often called the Southern Coastal Migration Route, pressed eastward, following the Indian Ocean coastlines through South Asia and Southeast Asia, and eventually reaching the landmass that is now Australia by approximately 50,000 years ago. This coastal route kept these populations at relatively low latitudes for much of their journey, where UV radiation remained intense and where dark skin remained strongly favoured. The indigenous populations of South Asia, the Andaman Islands, Papua New Guinea, and Australia preserve deeply dark pigmentation today. This is not because they have any special evolutionary relationship with dark skin that other groups lack. It is because they settled in environments where dark skin continued to be the most advantageous state, and the selection pressure to lighten never became dominant. Another branch of the migration pressed northward and westward, eventually reaching Western Asia and, from there, Europe. Parallel movements went into East Asia. These populations were entering environments of progressively lower UV radiation as they moved to higher latitudes, and the selective pressures acting on their skin color were beginning to change. But this change was not immediate, and it was not rapid by the standards of everyday human experience. Evolution acts across generations, not within lifetimes. The shift in selective pressure, from favouring dark skin to favouring lighter skin, had to accumulate over hundreds and thousands of generations before it produced a visible change in the population average. Research using population genetic modelling to estimate the timing of selective sweeps in European pigmentation genes suggests that the earliest of the major lightning-associated genetic sweeps began perhaps 30,000 years ago with one genetic variant, while the major sweeps in the genes most strongly associated with contemporary European paleness began only between 11,000 and 19,000 years ago. And even then, the change was not complete for thousands more years after that. This means that the tens of thousands of years between the first arrival of modern humans in Europe and the period when lighter skin began actively spreading were inhabited by people who were essentially as dark-skinned as their African ancestors had been. Hunter-gatherers in the forests and grasslands of a continent still emerging from the most recent ice age. And skilled at extracting resources from their environment, cognitively fully modern, culturally sophisticated in ways that the archaeological record has revealed with increasing richness, but dark-skinned for tens of thousands of years. Dark-skinned. The question is why? Why did the lightening take so long? Why did these people persist with dark skin in northern environments for so many thousands of years before the genetics began to shift? The answer has to do with diet, with the extraordinary nutritional richness of the hunter-gatherer lifestyle, and with the way that dietary vitamin D can substitute, at least partially, for UV-driven vitamin D synthesis. Section 6. Hunter-gatherers, diet, and the vitamin D safety net. Before the full story of skin lightening can be told, it is necessary to understand a factor that is often overlooked: the extraordinary nutritional wealth of the Mesolithic hunter-gatherer diet and the way that wealth provided a buffer against the vitamin D pressure that low UV northern environments imposed. Hunter-gatherers in Europe, particularly those living near coastlines, rivers and lakes, had access to food sources that are among the richest natural sources of vitamin D that exist anywhere in the food supply. Oily fish, salmon, herring, mackerel, trout, eel, caught in abundance from the rivers and coastal waters of Mesolithic Europe, are packed with vitamin D in their fat tissues. Wild game, particularly organ meats such as liver, and the fatty deposits of large mammals, also contain meaningful vitamin D. A hunter-gatherer regularly eating fatty fish and wild game could maintain reasonably adequate vitamin D levels even at northern latitudes, even with relatively dark skin that blocks more UV than light skin would. Examination of the bones of Mesolithic hunter-gatherers from European sites has found relatively low rates of the skeletal conditions associated with vitamin D deficiency, such as rickets, compared to what would later appear in farming populations. The hunters were dark-skinned, living at latitudes where UV synthesis through dark skin during the winter was limited, but their diet was compensating. They were getting vitamin D from their food rather than from the sun. This is not a minor point. It helps explain something that would otherwise be puzzling. If low UV at northern latitudes creates such strong selection for lighter skin, why did the dark-skinned hunter-gatherers persist without lightening for tens of thousands of years? The answer is that the selection pressure for lighter skin was partially neutralized by dietary compensation. It was present, accumulating quietly in the background, pushing the genetic frequency of lighter skin variants slowly upward over millennia. But it was not strong enough during the hunter-gatherer period to drive a rapid and complete population-wide shift. That changed when farming arrived. The Neolithic agricultural revolution, beginning around 10,000 to 11,000 years ago in the Fertile Crescent and spreading gradually into Europe over the following several thousand years, brought with it a dramatic transformation of the human diet. Cereal grains, wheat and barley and millet and rye, as became the staple foods of farming populations. Domesticated animals provided meat and dairy, but in compositions and quantities very different from the wild game of hunter-gatherers. And crucially, the new agricultural diet was dramatically poorer in vitamin D than the hunter-gatherer diet it replaced. Grains contain essentially no vitamin D. The domesticated animals of early farming communities, cattle and sheep and goats managed primarily for meat and dairy, provided substantially less vitamin D than fatty fish or organ meats from wild game. Early European farmers, analysing the evidence from their bones, show clear signs of increased vitamin D deficiency compared to the hunter-gatherers who preceded them. The agricultural revolution, whatever its benefits in terms of feeding larger populations, was a nutritional step backward in important ways. And this dietary change struck directly at the buffering mechanism that had allowed dark-skinned people to survive at northern latitudes without strong selection pressure toward lighter skin. Remove the dietary vitamin D safety net, and suddenly the UV-limited sun of northern Europe becomes the only option for vitamin D synthesis. And dark skin is bad at UV-limited vitamin D synthesis. The selection pressure for lighter skin, which had been building slowly for tens of thousands of years, suddenly became dramatically stronger with the shift to grain-based agriculture. This is one of the reasons researchers now believe that most of the actual lightening of European skin happened relatively recently, in conjunction with the spread of agriculture, or rather than gradually from the moment humans first arrived in Europe. The arrival of farming changed the selective landscape in ways that accelerated genetic change. And then, on top of that accelerated pressure from agriculture, came the most dramatic population movement European prehistory has yet revealed. Section 7. The Steppe Peoples Arrive. The Bronze Age Transformation. Around 5,000 years ago, on the vast grassland plains stretching from the northern shores of the Black Sea and the Caspian eastward into what is now Russia and Kazakhstan, a culture was developing that would reshape the genetic landscape of Europe and Asia more dramatically than any other event in human prehistory, outside the original out-of-Africa migration itself. These were the Yamnaya, so pastoralist herders who had developed a set of technologies that together gave them a mobility and a range of action, unlike anything that had existed in the human world before. They had domesticated the horse not just as food, but as a riding and working animal. They had developed the wheeled wagon, which meant they could carry substantial loads across enormous distances. They organized their entire economy around the herding of cattle, sheep, and horses across the steppe, moving with their animals, occupying the vast grasslands that were essentially inaccessible as permanent living territory to settled farming cultures. The Yamnaya were themselves a relatively recently formed population, the product of admixture between two ancestral groups. eastern hunter-gatherers from the forest steppe of Eastern Europe, and Caucasus hunter-gatherers from the highland regions between the Black and Caspian Seas. These two groups had come together genetically on the Pontic steppe sometime around 5000 BCE, producing a genetically distinctive population that carries a recognizable signature in ancient DNA that researchers have learned to identify with remarkable precision across samples from across Eurasia. Beginning around 3000 BCE, something shifted. The Yamnaya, or closely related steppe populations, began to expand outward from their homeland with what the ancient DNA record suggests was extraordinary speed and far-reaching extent. They moved westward into Central and Northern Europe. They moved eastward across the Ural Mountains into Central Asia. Their genetic signature appears in Bronze Age archaeological sites from Ireland in the west to the Altai Mountains of Mongolia in the east. The ancient DNA studies that first fully revealed the scale of this expansion, published in Nature in 2015 by two research groups working independently, one led by Johannes Krauss at the Max Planck Institute and one led by David Reich at Harvard, caused genuine scientific shock. The data showed that the genetic ancestry of Central and Northern European populations had shifted profoundly between the Neolithic and Bronze Age periods. People buried in corded ware culture sites, which spread across Northern Europe beginning around 2800 BCE, showed massive proportions of Yamnaya-related steppe ancestry, in some cases accounting for the majority of their total genetic heritage. This was not a slow cultural diffusion. This was large-scale population movement that fundamentally replaced the people who had been living there. The scale of the demographic transformation is difficult to fully appreciate without some concrete numbers. In some Northern European populations, Yamnaya-related steppe ancestry eventually accounted for 50 to 75% of total ancestry. Populations that had previously been predominantly descended from Neolithic farmers and Mesolithic hunter-gatherers were, within the span of a few hundred years on the genetic timescale, transformed into populations predominantly descended from steppe herders. The replacement appears to have been particularly pronounced in the male lineage. As the Yamnaya-related Y chromosome haplogroups came to dominate many European populations while pre-existing female mitochondrial lineages were partly retained. What this means in terms of the actual social dynamics of the period is intensely debated, but the genetic outcome is unambiguous. Along with the Yamnaya genetic ancestry came pigmentation variants. Ancient DNA analysis of Yamnaya individuals and their cultural descendants found that they carried genes associated with lighter skin pigmentation at elevated frequency compared to either the Mesolithic hunter-gatherers or the Neolithic farmers they encountered. In particular, genetic variants in the SLC45, A2 gene and contributions to the SLC24, A5 variant appear to have been carried at higher frequency in steppe-related populations and to have spread through Europe along with the broader Yamnaya expansion. The combination of all these forces, the pre-existing but slow accumulation of lighter-skin genetics through Neolithic farmer migration, the dramatically strengthened selection pressure from cereal-based agriculture, and the massive influx of new steppe-related ancestry carrying additional lighter skin variants, created the conditions for the final transition. Over the Bronze Age and Iron Age, roughly from 3000 BCE to the beginning of the historical period around 1000 BCE, the frequency of light skin alleles in European populations rose rapidly toward the high levels seen in modern Europeans. By approximately 3,000 years ago, pale skin had become dominant across most of the continent. The people who built the early Greek city-states, who fought in the Punic Wars, who constructed the great monuments of the Hallstatt and La Tène Iron Age cultures across Central and Western Europe, were living in a Europe that had recently, in evolutionary terms, completed this transition. They would have looked broadly similar to modern Europeans in terms of skin tone in many regions, though the full range of modern European paleness may not have been quite as widespread then as it is today. The Roman Empire itself would have been a place of considerable variation. Roman soldiers came from across the Mediterranean world and beyond. Their skin tones would have reflected the varied ancestries of a diverse empire, not the relative uniformity of pigmentation that characterizes modern northwestern European populations. It was only in the centuries after Rome's decline that the genetic and demographic picture settled into something approximating what we see in modern European populations. Section 8. The genetics in detail. Decoding what changed and why. To understand fully what happened at the molecular level, it helps to know something about the specific genetic changes that drove the lightening of skin in European and other populations outside the tropics. The science here has advanced dramatically in the past 2 decades, driven by genome-wide association studies in modern populations and by the explosion of ancient DNA analysis that has made it possible to watch genetic changes happening in real time across the archaeological record. So our human skin color is a polygenic trait. It is controlled not by one or two genes, but by dozens, perhaps more than 100, each contributing a relatively small independent effect to the overall phenotype. By current estimates, the genes studied in the most detail account for perhaps 60 to 70% of the heritable variation in skin color, with the remainder spread across many smaller effect variants. The polygenic nature of the trait is one reason it can respond so flexibly to natural selection. There are many genetic levers available, and selection can simultaneously push on multiple ones at once. Among the genes with the largest individual effects, SLC24A5 occupies a special position in the story of European skin lightening. This gene encodes a protein called NCKX5, a transporter that regulates the movement of sodium, calcium, potassium, and chloride ions across the melanosomal membrane. The specific activity of this transporter influences the chemical environment inside the melanosome and thereby affects the efficiency of eumelanin production there. One particular variant in this gene, referred to as the A111T variant, or by the genetic catalog identifier RS1426654, significantly reduces eumelanin synthesis when present. This single variant accounts for somewhere between 25 and 38% of the total pigmentation difference observed between European and sub-Saharan African populations. It is the most consequential single genetic change yet identified in the lightening of European skin. The A111T variant of SLC24A5 is found in nearly 100% of modern Europeans. It is also found at high frequency in populations of South Asian and Middle Eastern ancestry. In sub-Saharan African populations, it is essentially absent. The genetic evidence, including population genetic analysis of the region of the genome surrounding this variant, suggests it was under strong positive selection and spread rapidly through the populations that carry it. Research on the timing of this selective sweep suggests it began somewhere between 11,000 and 19,000 years ago, consistent with the Neolithic period and the spread of agriculture. The gene SLC45A2 encodes another melanosomal membrane transporter, and a variant in this gene, the L374F variant, also reduces eumelanin production and is strongly associated with lighter skin in Europeans. The timing of the selective sweep for this variant appears to be more recent than the SLC24A5 sweep, with the strongest selection apparently beginning around 5,800 years ago. This timing aligns with the Yamnaya migration and the Bronze Age transformation of Europe, suggesting this variant may have been particularly associated with steppe ancestry and may have spread through Europe on the back of the Yamnaya population expansion. The HERC2 and OC-A2 genes deserve mention for their role in eye color, which evolved somewhat independently of skin color. A regulatory variant in HERC2 reduces the expression of OC-A2, a gene that affects iris pigmentation, dramatically reducing the production of brown and black pigment in the iris and producing blue or green eye color. Yet ancient DNA analysis suggests this variant was already present in some Mesolithic hunter-gatherer populations in northern Europe, which is why individuals like Cheddar Man could have blue eyes while still having dark skin. Eye color and skin color While both aspects of overall pigmentation are governed by partly distinct genetic mechanisms and can diverge from each other over evolutionary time. Now here is one of the most beautiful demonstrations in all of evolutionary biology contained within this story. The problem of needing to produce less melanin in a low UV environment was solved not just once in Europe, but independently and simultaneously in East Asian populations as well. People from East Asia also inhabit higher latitude environments with reduced UV radiation. And they also have considerably lighter skin than equatorial African populations. The problem is the same. But when researchers examined the specific genetic changes responsible for lighter skin in East Asian populations, they found something striking. East Asian populations largely do not carry the same variants that drove skin lightening in Europe. The SLC24A5A111T variant, essentially universal in Europeans, is rare or absent in most East Asian populations. The SLC45A2L374F variant is similarly absent. Instead, East Asian populations carry different genetic variants that reduce pigmentation through different molecular mechanisms. The OCA2 gene, which has variants affecting pigmentation in both European and East Asian populations, shows evidence of independent selective sweeps in the two populations acting on different alleles within the same gene. Other loci associated with lighter skin in East Asians, including DDB1 and TMEM138 and others, are not similarly associated with lighter skin in Europeans. This is convergent evolution. Two populations, separated by thousands of miles and by an entire continent of diverse populations, facing the same selective pressure from reduced UV radiation at higher latitudes, independently evolved toward lighter skin through different genetic pathways. The destination was similar. The routes were completely different. Convergent evolution of this kind is powerful evidence that the selective pressure driving the change is real and strong. When the same environmental challenge independently produces the same biological outcome in two separate populations that arrived there through different genetic mechanisms, it argues strongly that natural selection, rather than chance or neutral drift, is responsible. The parallel paths to lighter skin in Europe and East Asia are one of the clearest examples of convergent adaptation to a shared selective environment in all of human evolutionary biology. Section 9. Modern health and what the ancestral story means today. The evolutionary history of human skin color is not only a story about the past. It has direct and practical implications for human health in the present, implications that affect millions of people's lives in concrete, measurable ways. Because skin color evolved as an adaptation to specific UV levels at specific latitudes, people whose ancestral biology was calibrated for one UV environment, but who now live in a very different one face health challenges that are, in the most literal sense, evolutionary mismatches. The body evolved for one set of conditions. It now finds itself in another. And the consequences of that mismatch are real. The most widely recognized mismatch involves vitamin D. Deeply dark-skinned individuals living at high northern latitudes, particularly in environments where significant time is spent indoors, in offices and schools and climate-controlled spaces, receive dramatically less UV exposure than ancestral populations at those latitudes would have received even with lighter skin. The combination of dark skin that blocks UV and reduced time outdoors in already low UV environments can make adequate vitamin D synthesis essentially impossible for significant parts of the year. The health consequences of chronic vitamin D deficiency are well documented and serious. Rickets, the bone disease of childhood caused by inadequate mineralization, is still diagnosed in dark-skinned children in northern countries at rates that reflect this mismatch. Vitamin D deficiency is a known risk factor for tuberculosis, and historical epidemiological patterns of tuberculosis mortality in dark-skinned populations in northern cities partially reflect this biological vulnerability. More recently, research has linked vitamin D deficiency to elevated rates of multiple sclerosis, type 1 diabetes, cardiovascular disease, various cancers, and complications during pregnancy and childbirth. None of these connections are speculative. They are supported by robust evidence from population studies and increasingly from clinical trials. The mirror image mismatch involves pale-skinned populations in high UV environments. Light skin, evolved to maximize UV penetration at low UV latitudes, is in high UV tropical or subtropical conditions profoundly vulnerable to UV damage. The dramatic migration of pale-skinned European populations to Australia, South Africa, and other high UV regions during the colonial era produced one of the sharpest mismatches in human evolutionary biology, and the health consequences have been severe. Australia has the highest rates of melanoma in the world. As South Africa and other regions with large pale-skinned populations at low latitudes also show dramatically elevated skin cancer rates. The folate-depleting effects of intense UV exposure on pale skin are also real, though less studied than the skin cancer consequences. Understanding these mismatches has practical implications for public health policy, for dietary recommendations, for guidance around sun exposure and sun protection, and for understanding health disparities that have sometimes been attributed to other causes. A dark-skinned person living in a northern city is not simply getting less sun. They are living in an evolutionary mismatch between their biology and their environment. And the medical response, which often involves vitamin D supplementation, is essentially a technological correction for a biological equation that nature never anticipated. This is not an abstract or theoretical concern. It affects the health of real people in meaningful ways, every day, in every northern city, and every high U5 region of the world, where migration has placed people in UV environments their bodies were not calibrated for. Section 10. What remains to be learned? The story is still being written. Ancient DNA research is still in its early decades, and the picture of ancient human pigmentation continues to grow clearer and more detailed with each new study. What we know today is already far more detailed and far more surprising than anything scientists could have told you about this topic 20 years ago. What we will know in another 20 years will likely be more surprising still. There are large regions of the world where ancient DNA research on pigmentation is still in its early stages. South Asia, Southeast Asia, the Americas, and much of Africa remain underrepresented in the ancient genome databases that have produced the clearest pictures of European pigmentation history. As more ancient genomes are recovered and analysed from these regions, the picture of global skin colour evolution will become more complete. There are also aspects of the story that remain actively debated. The precise relative importance of UV vitamin D selection versus other possible selective pressures, including protection against skin cancer, sexual selection, or adaptation to cold stress, continues to be evaluated. The specific timing of selective sweeps in different populations is being refined as more ancient genomes are analysed. The role of Neanderthal genetic contributions to early modern human adaptation in Europe is still being assessed, and there are questions that go beyond genetics altogether. The cultural and social dimensions of skin colour, how ancient people perceived it, how they responded to variation in it, whether it played any role in social organisation or in the forms of symbolic behaviour that increasingly show up in the archaeological record, are questions that genetics alone cannot answer. The biological story and the cultural story are intertwined, even if they operate on different timescales and through different mechanisms. What is already clear is that the science of skin colour evolution represents one of the most illuminating convergences of genetics, archaeology, physiology, and evolutionary biology that has yet been achieved. It has overturned century-old assumptions. It has revealed a past that is more diverse, more dynamic, and more surprising than anyone imagined. And it has shown, with clarity that was not available even a decade ago, that the visible differences between people are genuinely recent, genuinely responsive to environment, and connected through unbroken chains of ancestry to a single shared origin. Section 11. Africa's own diversity. The full spectrum at home. One thing that the simplest version of the skin colour story tends to obscure is the extraordinary diversity of skin pigmentation within Africa itself, a continent that is often incorrectly discussed as uniformly dark-skinned. Africa is the world's second largest continent, stretching from the Mediterranean coast in the north to the Cape of Good Hope in the south. From the Atlantic coast in the west to the Red Sea and Indian Ocean in the east. Within its borders lie some of the world's most intense UV environments and some of its more moderate ones. And the skin color of African populations varies accordingly, in ways that beautifully illustrate the same UV adaptation logic that applies globally. The darkest-skinned people anywhere in the world are found among the Nilotic and Nilo-Saharan-speaking pastoralists of East Africa, including groups like the Dinka and Nuer of South Sudan, who live near the equator under extraordinarily intense UV radiation, and who carry the highest levels of eumelanin pigmentation yet measured in any human population. Their skin is not simply dark. It is, by any biological measure, at the maximum expression of the protective eumelanin system that evolution has produced in Homo sapiens. The San people of southern Africa, sometimes called the Bushmen or Khoisan, who live in the Kalahari region at southern latitudes that receive considerably less UV radiation than the equatorial belt, have lighter skin than most other sub-Saharan African populations. Some San individuals carry variants in the SLC24A5 gene that are associated with lighter skin in European populations, which appears to have reached the San through relatively recent migration from East Africa rather than representing an independent evolution. Their skin tone, while darker than modern Europeans, is lighter than many other African populations. And the variation follows the UV gradient within the continent in exactly the way the vitamin D folate hypothesis would predict. Ethiopia and the Horn of Africa show considerable variation in skin color that reflects both latitudinal UV variation and complex population history involving ancient admixture between different African and Near Eastern populations. The Saharan and North African populations are generally somewhat lighter than equatorial Africans, again, consistent with their somewhat lower UV environments. The diversity within Africa is important for several reasons. It demonstrates that the same evolutionary principles apply everywhere, that skin color anywhere is a response to UV environment rather than an ethnic essence. It also complicates any simple narrative that treats dark skin as a single undifferentiated state that either exists or does not. The spectrum of African skin color is not uniform. It is as finely calibrated to local conditions as skin color anywhere else in the world. Section 12. The three-wave synthesis. How modern Europeans were made to draw the European story together into its fullest form, it is useful to understand the framework that geneticists call the three-source or three-population model for the origin of modern European genetic ancestry. This model has emerged from the synthesis of ancient DNA research and represents our best current understanding of how modern Europeans came to be genetically and phenotypically who they are. The first source is the Western hunter-gatherers, the dark-skinned, often light-eyed Mesolithic people who inhabited Europe from roughly 45,000 years ago, when modern humans first arrived on the continent, until the Neolithic period. Their contribution to modern European ancestry is estimated at roughly 10 to 30% in most European populations, with somewhat higher proportions in some Scandinavian and Iberian populations where they may have persisted longer before being absorbed by incoming farmers. Their characteristic genetic signature, including the Mesolithic pigmentation pattern of dark skin with light eyes, still flows through modern European genomes in these proportions. The second source is the early European farmers, Neolithic people descended from populations in Anatolia who developed agriculture in or near the Fertile Crescent, and whose descendants spread westward through the Balkans and across Europe beginning roughly 9,000 years ago. These populations carried a different genetic makeup than the hunter-gatherers, including higher frequencies of the SLC24A5 light-skin variant, and their migration introduced this variant into Europe's gene pool more broadly. In most European populations, early European farmer ancestry contributes perhaps 20 to 50% of total ancestry, with higher proportions in southern Europe and lower in the far north, where the Yamnaya influence was strongest. The third source is the Western steppe herders, descended from the Yamnaya and closely related cultures. Their massive expansion into Europe beginning around 3000 BCE fundamentally reshaped the genetics of Central, Northern, and Eastern European populations. In many Northern European populations, steppe-related ancestry accounts for 50% or more of total ancestry. Along with the broad steppe genomic contribution came additional pigmentation lightning variants, the genetics for lactase persistence, and many of the ancestral lineages associated with modern Indo-European languages. The interplay of these three genetic streams, combined with thousands of years of ongoing natural selection for lighter skin driven by the agricultural diet change and the low UV northern environment, produced the modern European genetic landscape. Each wave brought different pieces of the pigmentation puzzle. Selection then drove the light skin variants, whatever their specific source, toward the high frequencies they have reached in modern populations. This is why we should not look for a single simple moment or a single simple cause for the paleness of modern Europeans. There was no single mutation that suddenly made Europeans pale. There was no single migration that completed the transformation. There was instead a long, complex, multiply-sourced process of population mixing and natural selection, unfolding over thousands of years, producing through that long process an outcome that none of the participants could have anticipated or intended. The paleness of modern Northern Europe is the unplanned product of ancient herders and ancient farmers and ancient hunters, of solar radiation and vitamin D and folate, of ice ages and population bottlenecks, and the accident of which migrations happened when. It is as contingent and as surprising as all of evolutionary history, which is to say, very. Section 13. Closing. The long arc of a quiet story. It is late now. The kind of late where the world outside has gone still, and the only sounds are the soft ones that you stop noticing after a while. The gentle background of a house at rest, the distant hum of something mechanical and unintrusive, the sound of your own breathing, slow and steady as something sleeping. We have traveled a long way tonight, further than any single night should probably go, and yet not nearly as far as the story deserves, because the story deserves all of time. We began with a question that seems simple but turns out to be anything but. How did humans become white? How did a species that began uniformly dark-skinned under the equatorial African sun come to include in its number people as pale as the palest Scandinavian, as fair as the most light-skinned person from any tradition that has ever existed on Earth? And the answer, traced carefully through the evidence that science has accumulated, is this. It begins approximately 1.2 million years ago, when our ancestors lost their body hair in the service of staying cool during the extended physical exertion of life on the open savanna. That hairlessness exposed pale skin to intense equatorial UV radiation, and dark eumelanin-rich skin evolved in response, protecting the folate that those ancestors needed for healthy reproduction. Dark skin became the ancestral state of the species, the starting point from which all subsequent variation would eventually develop. Then, beginning perhaps 60,000 to 70,000 years ago, small groups of those dark-skinned ancestors began walking out of Africa. They followed coastlines and river corridors into a wider world. They eventually reached every habitable landmass on Earth, and wherever they went, their bodies began the slow process of adjusting to the UV conditions of their new environments, though that process was far slower and far less complete during the hunter-gatherer period than anyone previously assumed. In Europe, the dark-skinned Western hunter-gatherers persisted for tens of thousands of years, buffered from strong selection for lighter skin by diets rich in vitamin D from wild fish and game. Their bodies maintained dark skin and, in many cases, light eyes, a combination that the ancient DNA evidence has revealed with a clarity that surprised the world when it was announced. Then farming arrived from Anatolia, bringing Neolithic farmers with their somewhat lighter skin genetics and their grain-based diets that stripped away the dietary vitamin D safety net. The selection pressure for lighter skin intensified. Some lightning occurred, But the process was still incomplete when the Yamnaya herders exploded out of the Eurasian steppe around 5,000 years ago, bringing their horses and their wagons and their genes and reshaping the genetic landscape of Europe in ways that are still being fully understood. The combined effect of all these forces, played out over thousands of years of ongoing natural selection, finally pushed pale skin to dominance across most of Europe roughly 3,000 years ago. The iconic pale European, the feature so often imagined as ancient and timeless, arrived in evolutionary terms almost yesterday. This story does not diminish anyone. It does not elevate anyone. It does not create a hierarchy where none existed before. What it does is connect everyone. It shows that the extraordinary range of human skin tones visible in the world today is not a collection of fixed essences, ancient and immutable, expressing fundamental differences between populations. It is a collection of recent adaptations, each one a biological response to a specific UV environment, each one traceable through chains of migration and selection, back to the same dark-skinned African ancestors that every person on Earth shares. While every skin tone is the right answer to the question asked by the particular geography where those genes were shaped. The deep black of equatorial East Africa is the right answer to the question of life under the most intense UV radiation on Earth. The pale skin of northern Scandinavia is the right answer to the question of vitamin D synthesis under the weakest northern sun. The brown of South Asia, the olive of the Mediterranean, the reddish tones of indigenous Americans, the varied browns of Southeast Asia and the Pacific, each one is the right answer to a different question asked by a different geography across a different stretch of deep time. Skin color is geography made visible in cells. It is sunlight expressed in molecules. It is the memory of places written into bodies by the patient arithmetic of generations beyond counting. And it connects every one of us, whatever we look like, through unbroken lines of descent, all the way back to the beginning, to the dark-skinned people who first stood in the open air of Africa and squinted into the equatorial sun. That is your ancestry. That is everyone's ancestry. The long walk, the slow change, the invisible work of evolution across all of those years, producing from a single starting point the full, extraordinary, still astonishing diversity of the human species. You carry that whole story in your skin. Sleep well tonight. Rest deep. And when morning comes and you catch your reflection, remember that whatever color looks back at you, it is the answer to a very old question. And the question is simply, where did we live and what did the sun ask of us there? If tonight's journey through deep time stirred something in you, please consider leaving a like and subscribing to History for Sleep. Every interaction genuinely helps this channel continue creating these long, carefully researched explorations of the science and history that shape the world we inhabit. If there is a topic you would like to hear explored in this slow and careful way, please leave it in the comments below. Some of the most rewarding journeys on this channel have started exactly there, from a question left in the quiet of a late night, just like this one. Thank you for spending this time here. Rest well. Research for this script draws on published work from Nina Jablonski and George Chaplin at Penn State University. Ancient DNA studies from the Natural History Museum, London, University College London, Harvard University, the Max Planck Institute for Evolutionary Anthropology, and the University of Ferrara. Studies published in Science, Nature, PNAS, BioRx14, and peer-reviewed journals in population genetics and human evolutionary biology, and the established literature on pigmentation genetics as of 2025. Section 14. The Ice Ages and their influence. Cold darkness and the pressure that built. To fully appreciate what those dark-skinned Western hunter-gatherers were enduring in Northern Europe, it helps to understand something about the climate conditions under which they lived. Conditions very different from anything that exists in Europe today, and conditions that would have had their own complex relationship with the UV pressures we have been discussing. Modern Europe, whatever its temperatures and seasonal variations, is a relatively benign environment compared to what it was during and immediately following the most recent glacial maximum, the period roughly 26,000 to 19,000 years ago, when the ice sheets of the last ice age reached their greatest southern extent. At the glacial maximum, an enormous ice sheet covered most of what is now Britain, Scandinavia, and northern continental Europe. The remaining unglaciated parts of Central and Southern Europe were tundra and grassland, cold and windswept, inhabited by woolly mammoths and cave lions and giant deer and the bands of human hunters who pursued them. UV radiation during these periods was not dramatically different from what it is today in terms of the basic physics of UV delivery, but the behavioral responses to cold, the need for heavy clothing, the construction of shelters, the spending of significant time in enclosed spaces would have substantially reduced the skin surface area exposed to sunlight. Even weak winter UV cannot drive vitamin D synthesis through clothing. The very conditions that produced strong selection for lighter skin, the combination of weak UV and reduced exposure from cold weather clothing and sheltering behavior, were also the conditions that made existing in northern latitudes possible at all. The populations that managed to survive in Europe during the glacial maximum and the period immediately following were small. Population genetic modelling suggests that European populations went through severe bottlenecks during the cold periods, with numbers declining to very low levels in refugia in southern Europe and the Balkans before expanding again as the climate improved. These bottlenecks are relevant to the pigmentation story because they would have reduced genetic diversity and potentially allowed genetic variants to drift in ways not purely driven by selection. After the last glacial maximum, beginning roughly 19,000 years ago, the ice sheets began to retreat. Europe gradually warmed, forests spread northward, and the vast herds of megafauna that had characterized glacial Europe were either adapting or declining, eventually leading to the extinctions of many species over the following millennia. Human populations began to expand again, moving northward into territories that had been covered by ice, following the retreating tundra and the animals that depended on it. The people who repopulated northern Europe during this post-glacial expansion were carrying the dark-skinned genetics we have discussed. They were adapting to an environment that was both geographically familiar and, in its UV and dietary characteristics, creating selection for lighter skin. But the selection was slow, and the buffering from their rich hunter-gatherer diets remained effective enough that the change accumulated gradually rather than dramatically. It was only with the combination of later agricultural transition and Yamnaya migration that the rate of change accelerated to the point where the shift became visible in the archaeological genetic record within a span of a few thousand years rather than tens of thousands. This pattern of slow background accumulation, followed by rapid acceleration, is not unique to the European skin lightening story. It appears repeatedly in evolutionary biology. Selective pressures can build over long periods, with the genetic variants being favoured only gradually increasing in frequency until some change in circumstances dramatically increases the intensity of selection and the change accelerates. The introduction of agriculture, by removing the dietary vitamin D safety net that had buffered dark-skinned hunter-gatherers for tens of thousands of years, appears to have been precisely such a change in circumstances, converting a slow background pressure into an acute one. Understanding this temporal pattern helps make sense of the ancient DNA evidence. The presence of dark-skinned individuals in Mesolithic Europe for tens of thousands of years does not mean selection for lighter skin was absent. It means the selection was weak enough to be offset by dietary compensation. The sudden relative rapidity of the change during the Neolithic and Bronze Age does not mean something dramatically new was discovered. It means the buffering mechanism was removed and the existing selection became dominant. Section 15. The Unexpected Eye. Blue eyes, brown eyes, and the story of iris. Pigmentation. The story of eye color in Europe is one of the more intriguing subplots in the overall narrative of human pigmentation, not least because it reveals how apparently connected traits can actually follow somewhat independent evolutionary trajectories. Blue eyes are, in the global context, extremely unusual. They are found predominantly in populations of European descent, with the highest frequencies in the Baltic region, Scandinavia, and the British Isles, where in some populations more than 80% of individuals have blue or grey eyes. Outside of Europe, blue eyes are rare, They occur at elevated frequency only in some populations of South Asia and Central Asia, and even there, they are minority features. The genetic basis of blue eye color was not fully clarified until the 2000s, when researchers identified that a substantial portion of blue eye color in Europeans traces to a single regulatory variant in the HERC2 gene. This variant, RS129138332, affects the expression of the neighboring OCA2 gene, which encodes a protein involved in regulating the pH of melanosomes in iris melanocytes. When OCA2 expression is reduced by the HERC2 variant, the melanosomes function less efficiently, producing less melanin in the iris, and the iris appears blue or grey rather than brown. Intriguingly, a population genetic analysis of the HERC2 variant suggests that it originated in a single individual somewhere around the Black Sea region, perhaps 6,000 to 10,000 years ago, and that all blue-eyed people alive today are descended from that one individual, or at least from a very small founding group carrying the same or similar variants. This is not to say that the ancestor of all blue-eyed people is easily identifiable or historically traceable. It simply reflects the population genetic reality that some genetic variants, when they are initially rare and spread primarily by drift and perhaps mild selection rather than intense adaptive pressure, can be traced back to relatively recent single origins. The ancient DNA record shows the HERC2 blue eye variant appearing in European populations during the Mesolithic period, most perhaps around 14,000 years ago or somewhat earlier in some analyses. Critically, it appears in individuals who still had dark skin, as demonstrated by the Cheddar Man example and other similar cases. This decoupling of eye color from skin color in the ancient European genetic record shows that these two aspects of overall pigmentation, while both involving melanin and melanocytes, evolved at least partly independently and under at least partly different selective pressures. The evolutionary advantage of blue eyes, if any, is much less clear than the advantage of lighter skin for vitamin D synthesis. Unlike skin color, which has a direct and mechanistically understood relationship to UV-dependent physiology, blue eye color has no confirmed strong adaptive advantage in northern environments. Suggestions have included that blue eyes might be better at processing low light conditions in dark northern winters, or that the iris pigmentation is genetically linked to other traits that were being selected for, or that sexual selection played a role, with blue eyes simply becoming fashionable in a population where they were rare and therefore striking. None of these hypotheses is as well supported as the skin color vitamin D connection. The most honest current summary of the science is that we do not fully understand why blue eyes spread through northern European populations. What the ancient DNA record does suggest is that light eye color was one of the first aspects of European pigmentation to change, preceding the lightening of skin color by thousands of years. The first light-eyed individuals appear in the Mesolithic, while light skin only began spreading with the Neolithic transition and later. This temporal disconnect between eye lightening and skin lightening supports the idea that different selective pressures were operating on these two aspects of the same overall pigmentation system. The Cheddar Man, dark-skinned and blue-eyed, represents a real and probably widespread phenotype among Mesolithic Western hunter-gatherers, individuals who had already experienced the early spread of light eye variants in their population, but whose skin remained dark because the selection pressure for lighter skin had not yet become dominant. Understanding this disentangles what might otherwise seem like a paradox and reveals it as simply two different biological systems responding to two different sets of pressures at two different rates. Section 16. The modern moment. Living in an evolutionary mismatch. There is a dimension to the story of human skin color that extends directly into the present day, not in any abstract historical sense, but in the practical terms of everyday health and disease in the modern world. When we talk about populations whose skin color was calibrated by evolution for one UV environment, but who now live in a very different one, we are not talking about rare edge cases or theoretical concerns. We are talking about hundreds of millions of people in the modern world, navigating the biological consequences of migrations that have moved them far from the environments where their pigmentation genetics were shaped. Consider the situation of South Asian populations who have migrated to the United Kingdom, Scandinavia, or other high-latitude northern European countries. Their pigmentation genetics were shaped by centuries or millennia of life in the Indian subcontinent, where UV radiation is substantially higher than in northern Europe. Their skin, while often described in general terms as brown rather than deeply dark, carries enough melanin to significantly reduce UV penetration. At the latitudes of London or Oslo or Stockholm, particularly during the winter months when UV radiation is weakest and day length is shortest, individuals with this level of pigmentation may produce essentially no vitamin D through skin exposure at all during several months of the year. This is not speculation. It is documented in vitamin D blood level studies across South Asian, East African, West African, and other dark-skinned immigrant populations in northern European countries. These populations consistently show rates of vitamin D deficiency that are substantially higher than those seen in the lighter-skinned indigenous populations of those countries. And vitamin D deficiency, as we have explored, is not a trivial condition. It is associated with bone disease, immune dysfunction, elevated cancer risk, pregnancy complications, and a range of other serious health outcomes. The public health response to this has been uneven. Some countries have implemented targeted vitamin D supplementation programs for populations at risk. Others have left individuals to manage the mismatch on their own, often without adequate awareness that a mismatch exists. Medical practitioners in northern countries have, over the past decade or two, must become increasingly aware of the vitamin D deficiency problem in dark-skinned populations, but implementation of routine testing and supplementation recommendations has been inconsistent. The reverse mismatch, very pale-skinned people living in high UV environments, has its own serious health consequences that have played out with historical clarity in Australia and similar regions. When large-scale European migration to Australia began in the late 18th century and accelerated in the 19th and 20th centuries, it placed populations with pigmentation genetics calibrated for the northern European environment into one of the highest UV environments on Earth. The results have been stark. Australia has the world's highest rates of skin cancer, including melanoma. The cultural response, the Slip Slop Slap sun protection campaign and the broad public awareness of sun protection in Australia represents in effect a cultural technology attempting to correct for an evolutionary mismatch. The evolutionary history of skin color also intersects with the story of vitamin D and disease in African American populations in the United States, who have been shown in multiple studies to have substantially higher rates of vitamin D deficiency than European American populations, and who also have historically higher rates of certain diseases that vitamin D deficiency can exacerbate, including tuberculosis, certain cancers, and preeclampsia in pregnancy. These health disparities have multiple causes, including social, economic, and healthcare access factors. But the biological mismatch component, a deeply dark skin calibrated for equatorial UV environments living at mid-latitude North American latitudes, is real and medically relevant. Understanding the evolutionary origins of skin color does not solve these public health problems by itself, but it provides an essential biological framework for understanding why they exist and what interventions might address them most effectively. A vitamin D supplementation program targeted at populations at risk makes evolutionary sense in a way that a generic recommendation for everyone equally does not. Medical awareness of which populations are most likely to be vitamin D deficient and why follows directly from understanding the UV adaptation logic of skin color evolution. Section 17. The question of sexual selection. Beauty, rarity, and the role of preference. In any discussion of skin color evolution that aspires to completeness, it would be an omission not to at least address the question of sexual selection, even though it is a topic considerably more contested and less well evidenced than the UV adaptation story we have explored in detail. Sexual selection, the process whereby traits become more common in a population because they are preferred by potential mates rather than because they directly improve survival or physiological function, is a real and significant force in evolution. It produced the peacock's tail, the lyrebird's extraordinary vocal mimicry, and the striking plumage of countless bird species. It almost certainly plays some role in human evolution, though the specific traits it has acted on and the magnitude of its effects are much debated. Some researchers have suggested that sexual selection may have contributed to the evolution of certain pigmentation features in human populations, particularly features that are not easily explained by UV adaptation alone. Blue eyes in Northern Europe, as we have discussed, have an unclear adaptive benefit that some researchers attribute at least partly to sexual selection operating on a rare and striking trait. Blonde hair, which is at highest frequency in Scandinavian and Baltic populations, may similarly involve some component of sexual selection, since it is difficult to identify a straightforward physiological advantage to hair lacking dark pigment. The cultural anthropologist and evolutionary biologist Peter Frost has proposed that sexual selection played a significant role in driving the high frequency of light eyes, light hair, and pale skin in Northern European populations, suggesting that these features became preferred partly because they were rare and therefore reliable signals of local ancestry, or because of direct aesthetic preferences that varied across cultures. This hypothesis remains controversial and is difficult to test rigorously with the available evidence. What the ancient DNA evidence does show is that light hair and light eyes in European populations have a somewhat different temporal and geographic history from light skin, with light eye variants appearing earlier and in different population contexts than the major skin lightening genes. If sexual selection played a role in the spread of light eyes or light hair, yet it would have been operating on a somewhat different timescale and through somewhat different mechanisms than the vitamin D-driven selection for lighter skin. The most honest assessment of the current evidence is probably this: UV adaptation, operating through the vitamin D folate mechanism, is the primary and best supported explanation for the lightening of skin tone in European populations and for the broad global patterns of skin color variation. Sexual selection may have contributed to some features of European pigmentation, particularly aspects like eye and hair color that are less tightly constrained by UV physiology, but its contribution is harder to quantify and less well evidenced. The dominant story is one of biological adaptation to varying UV environments, not one of aesthetic preference driving the changes we see. As Section 18, Africa's ancient diversity and the genetics of dark skin. We have spent considerable time on the story of skin lightening in non-African populations because that story contains many surprises and because it is the one that has been most dramatically rewritten by recent research. But it would be incomplete to leave the story without spending more time on the complexity of dark skin genetics in Africa itself, which turns out to be far more genetically diverse than was previously understood. For a long time, researchers focused on a relatively small number of genes when studying skin color evolution, and the implicit model was that dark skin was ancestrally simple, a single state maintained by selection in equatorial environments. The more recent and comprehensive genetic studies have revealed a picture that is considerably more complex and in some ways more interesting. A landmark study published in 2017 by Sarah Tishkoff's research group at the University of Pennsylvania examined thousands of Africans from ethnically diverse populations, measuring their skin color with reflectometry and analyzing their genomes in detail. They found that the genetic architecture of skin color in Africa is itself highly diverse and complex, with different genetic variants contributing to pigmentation in different populations across the continent. One particularly striking finding was that some of the genetic variants associated with lighter skin in non-African populations, variants that were thought to be recent derived mutations arising after the out-of-Africa migration, were actually present in ancient polymorphic form in African populations. This means that the genetic diversity for skin color within Africa is itself very old, predating the out-of-Africa migration, and that populations outside Africa were not starting from scratch genetically when selection for lighter skin began. They were in many cases selecting on pre-existing variation, amplifying variants that had been present in low frequencies in the ancestral African population. This finding also means that statements like all humans were once dark-skinned, while strictly true from the phenotypic standpoint, require some nuance from the genetic perspective. The ancestral African population was phenotypically dark, but it already contained some of the genetic variation that would eventually be amplified to produce lighter skin in dispersed populations. Selection could work relatively quickly in some cases, not because novel mutations had to arise, but because it was amplifying rare variants that already existed. Among the specific genes implicated in African skin color diversity, the study found novel roles for genes like MFSD12, OC-A2, HERC2, and DDB1/TMEM138 in varying the range of skin tones within Africa. These are genes that affect melanin chemistry and melanosome function in ways that create variation across the brown to very dark brown spectrum, rather than the brown to very pale spectrum that is more commonly discussed in the context of non-African populations. The Tishkov study also confirmed the extraordinary depth of genetic diversity within Africa, reflecting the fact that modern humans have inhabited the continent for far longer than they have inhabited anywhere else, and that African populations have had more time to accumulate and partition genetic variation across the continent's diverse UV environments. The pattern within Africa, darker skin near the equator and somewhat lighter skin at higher latitudes both north and south, is the same pattern seen globally, but expressed across a narrower range of pigmentation and across a population with much deeper genetic history. This African genetic diversity is also medically relevant. Many of the genetic variants associated with vitamin D metabolism and skin cancer risk that have been characterized primarily in European populations turn out to have African counterparts, and understanding the functional significance of African pigmentation variants is important for addressing health disparities in African and African ancestry populations around the world. Section 19. The Toba Catastrophe and Genetic Bottlenecks. How near extinction shapes evolution. One more piece of context for the out-of-Africa migration and the subsequent evolution of human pigmentation deserves discussion, because it may have had a profound effect on the genetic starting point from which all subsequent skin color evolution outside Africa proceeded. Around 74,000 years ago, a massive supervolcanic eruption occurred at what is now Lake Toba in Sumatra, Indonesia. The Toba eruption is one of the largest known volcanic events in the past 2 million years, and it injected enormous quantities of sulfur dioxide and ash into the upper atmosphere, causing what researchers call a volcanic winter, lasting perhaps several years, and a subsequent period of global cooling that may have lasted 1,000 years or more. The ecological and demographic effects on human populations are debated in the scientific literature, with some researchers arguing for a catastrophic near-extinction event that reduced the global human population to only a few thousand individuals, and others suggesting the effects were less severe, particularly in tropical regions with dense and resilient ecosystems. Now, the genetic evidence for a severe bottleneck around this time is consistent with the Toba hypothesis, but does not definitively prove it, since other population size reductions could have left similar genetic signatures. What is not disputed is that human populations outside Africa appear to have passed through at least one severe genetic bottleneck somewhere in the period around 50,000 to 100,000 years ago, a bottleneck that dramatically reduced genetic diversity relative to African populations, and that established the relatively low diversity characteristic of all non-African human populations today. This bottleneck means that the founding populations of Europe, Asia, and the Americas were small, genetically impoverished relative to their African contemporaries, and carrying only a subset of the genetic variation present in Africa at the time. This has implications for the skin color story. The small founding populations would have carried only a limited sample of the full range of pigmentation variants present in Africa, and genetic drift in those small populations could have fixed or dramatically shifted the frequency of some variants, relative to their frequency in the African source population. Whether this contributed to the starting conditions for lighter skin evolution in non-African populations is an open question in the field, but it is a dimension of the story that the simplest telling often omits. The bottleneck also helps explain one of the features of non-African pigmentation genetics that would otherwise be puzzling. Why the specific genetic variants driving lighter skin in Europeans and East Asians are different from one another, despite both populations descending ultimately from the same out-of-Africa founding group. If the founding population of the out-of-Africa migration was small and carrying only a limited genetic toolkit, and if that population then split into the ancestors of Europeans and Asians at a further bottleneck, each subsequent founding population would have had slightly different genetic variants available for selection to act on when the pressure for lighter skin began. The result is convergent phenotypes achieved through divergent genetics, exactly the convergent evolution pattern we have explored. As Section 20, a final reflection on time, skin, and what it means to be human. As we near the end of this long journey through deep time and evolutionary biology, it seems worth pausing on something that the story reveals not just about skin color, but about the nature of being human more generally. We are a young species with an old body. Modern Homo sapiens has existed for perhaps 300,000 years, which is young by the standards of many mammal species. But the biological systems we carry, the melanin production machinery, the vitamin D synthesis pathway, the reproductive physiology that made both of these things evolutionarily important, are far older than that. They evolved over millions of years of earlier hominin history. And they were refined during those hundreds of thousands of years of exclusively African existence before the migration began. When our ancestors walked out of Africa 60,000 to 70,000 years ago, they were carrying a set of biological tools calibrated for one set of conditions. Over the tens of thousands of years that followed, as they spread into environments very different from the equatorial Africa those tools were calibrated for, evolution slowly recalibrated. But evolution is patient in ways that individual lives are not. The recalibration happened across hundreds of generations, not within any single person's lifetime. Every individual in every generation was simply living, simply surviving, simply trying to feed themselves and their children and get through the winter. None of them knew they were part of an evolutionary story. But none of them was aware that the slight advantage their lighter skin conferred in a low UV environment was incrementally shifting the genetic composition of the population they belonged to. This is one of the things that makes evolutionary biology both humbling and illuminating. The most visible feature of individual humans, the one that has been the subject of more cultural and social significance than perhaps any other biological trait, the one that has been used to construct elaborate and often deeply harmful hierarchies and categorizations. This feature turns out to be nothing more than a relatively recent, still changing, geographically variable adaptation to ultraviolet radiation levels. It is calibrated to the latitude where your ancestors happened to live. It is no more or less fundamental to who you are than the shape of the earlobe or the curvature of the fingerprint. And it is changing, not quickly, not in any way any of us will witness. But the selective pressures that shaped human skin color over the past tens of thousands of years are still operating today, in the modified and muffled form that modern life imposes. Populations that have migrated to new UV environments are, generation by generation, in those long, quiet timescales that evolution uses, beginning the same process of slow recalibration that has happened every time humans have moved to a new place. The past is not a fixed thing. It is a continuous process that runs through the present into the future. The story of skin color is not over. It is still being written in the bodies of everyone alive, on in the choices being made by natural selection across every generation, in the ongoing dialogue between human biology and the light of the sun. We are all, in our skin, works in progress. Adaptations still adapting, responses still responding. The long story of what it means to be human, written in the most visible surface we have, carrying on. That is not a small thing to carry. It is, perhaps, a beautiful one. Section 21. The complexity we did not expect. Repigmentation, reverse migrations, and the flexibility of skin color. One of the most counterintuitive findings in the modern study of skin color evolution is that the story has never simply been one of darkening in equatorial regions and lightening elsewhere, with each population moving in only one direction from a common dark starting point. The reality, revealed by both modern population genetics and ancient DNA, is considerably more complex and includes events of repigmentation, where populations that had become lighter subsequently evolved back toward darker tones as they moved into higher UV environments. Consider the case of the indigenous Polynesian peoples, who inhabit some of the most remote islands in the Pacific Ocean. These populations are derived from ancestors who originated in East Asia, populations that by the time of the Pacific migration had presumably undergone some degree of lightening due to their time at higher East Asian latitudes. Yet modern Polynesian populations are considerably darker than modern East Asians, reflecting the extreme UV environments of the tropical Pacific islands they settled. The reverse journey, from partial lightening back toward darker pigmentation, occurred over the relatively short period since the Pacific migration, estimated at only a few thousand years in the case of some island groups. This rapid repigmentation demonstrates that the selective pressure from UV radiation can drive pigmentation change in either direction with similar speed, depending on which direction is favored in the new environment. The genetic toolkit for adjusting melanin production is flexible in both directions. Variants that increase eumelanin production can be selected for when protection from intense UV is needed, just as variants that decrease it can be selected for when more UV penetration is needed for vitamin D synthesis. A similar pattern of repigmentation appears to have occurred in some African populations that had previously been at higher latitudes and underwent some lightening, then moved back into high UV equatorial zones. Researchers studying the genetics of the Maasai and other Nilo-Saharan pastoralists, who have some of the darkest skin tones measured anywhere in the world, have found evidence suggesting that the extreme darkness of their pigmentation reflects not merely the preservation of ancestral dark skin, but an enhancement of it, a further intensification of dark pigmentation in populations with deep ancestral histories involving movement into and out of high UV East African environments. These cases of repigmentation add yet another layer of complexity to the skin color story. And it is not a one-way journey from dark in Africa to progressively lighter elsewhere, with each population marching steadily from dark origins toward the pale extreme. It is a dynamic, ongoing calibration, with populations in different environments tuning their pigmentation toward whatever level is most adaptive for their current UV exposure, constrained by the genetic variation available in their lineages, but fundamentally responsive to environmental pressure in either direction. The practical implication of this flexibility is important. Human skin color is not fundamentally fixed by ancestry in the way that some historical thinking has assumed. It is a plastic adaptive trait that responds to selection over relatively short evolutionary timescales. This is part of why researchers estimate that a population can achieve close to optimal pigmentation for a new UV environment in roughly 10,000 to 20,000 years, or perhaps as few as 3,000 to 4,000 years if selection is intense. These are short timescales by the standards of evolutionary change in most complex traits, suggesting that selection on skin color has been unusually strong throughout human history. This flexibility also means that current skin color patterns across the world are probably not in complete equilibrium with their environments. Migration, medicine, clothing, and shelter all dampen the selection pressure that would otherwise drive ongoing adjustment. Modern dark-skinned populations in northern cities are not likely to become dramatically lighter over the next 1,000 generations, because the health consequences of their vitamin D mismatch, while real, are not sufficiently severe in a world of dietary supplementation and medical care to produce the kind of strong selective pressure that would rapidly shift allele frequencies. The evolutionary process is still running in the background, but more slowly than it would have in the ancestral world. Section 22. The role of agriculture in the northern and southern hemispheres. The story we have told about agriculture and skin color has focused primarily on Europe, because Europe is where the most detailed ancient DNA record exists and where the interaction of farming with pigmentation has been most thoroughly studied. But the same dynamics were playing out, with their own local variations. Saw wherever farming spread and wherever it brought populations into nutritional conditions that altered their vitamin D availability. In East Asia, the development of rice agriculture in the Yangtze River Valley and millet agriculture in the Yellow River Valley, beginning perhaps 10,000 to 9,000 years ago, similarly shifted populations away from the vitamin D-rich diets of hunter-gatherers towards cereal-based nutritional regimes. The selection pressure for lighter skin in East Asian populations, which appears to have acted through different genes than in Europe, was likely intensified by the same dietary dynamic. East Asian populations who were farming grain-based crops at higher latitudes would have faced the same reduction in dietary vitamin D that European farmers faced, creating similar selection pressure for increased UV penetration to compensate through skin synthesis. The parallel timing of skin lightening in European and East Asian populations, both of which appear to have undergone major selective sweeps for lighter skin variants within roughly the past 10,000 to 20,000 years, may reflect not just the common selective pressure from low UV at higher latitudes, but also the common dietary pressure from the independent development of grain agriculture in both regions. Agriculture, wherever it emerged, may have consistently accelerated the lightening of skin in populations living at latitudes where UV was marginal for vitamin D synthesis. In South Asia, a different pattern emerges. The Indian subcontinent spans a huge range of latitudes, from tropical extremes in the south to temperate conditions in the far north. Agriculture developed there both through independent domestication and through influence from the Fertile Crescent tradition. The pigmentation of South Asian populations shows enormous variation, from the very dark tones of some Dravidian populations in the far south to somewhat lighter tones in northern populations with more admixture from Central Asian and Iranian populations. The SLC24A5 variant that drives lightening in European populations is also found at high frequency in much of South Asia, having arrived through migrations from Western Eurasia, and it contributes to the somewhat lighter average skin tones in northern India and Pakistan compared to southern India and Sri Lanka. The Americas present a somewhat different story because agriculture there was developed independently rather than being imported, and the migrating populations who first entered the Americas through the Siberian crossing did so relatively recently on an evolutionary timescale, somewhere between 25,000 and 15,000 years ago, with the main wave of migration more recently than that. The indigenous people of the Americas show considerable variation in skin tone, reflecting both the UV environments of the regions they settled and the complex population history of the various migrations that peopled the hemisphere. The relatively recent nature of the Americas settlement means that adaptation to local UV conditions may not yet be complete, and the ancient DNA record for the Americas, still in early stages of development, may eventually reveal stories of pigmentation change parallel to those we have seen in Europe. Section 23. What ancient people knew and did not know about their own skin. Sitting with the immensity of this story, spanning hundreds of thousands of years and involving forces entirely invisible to the people who experienced them, it is worth pausing to think about what any of this meant to the actual people who were living through it. The Mesolithic hunter-gatherer with dark skin and blue eyes, ranging through the forests and wetlands of what is now England 10,000 years ago, did not know he was dark-skinned in any categorical sense that we would recognize. There was no concept available to him of a hierarchy of skin tones, so no cultural framework for comparing his complexion to that of people far away in Africa whom he had no knowledge of. His appearance was simply his appearance, the appearance of everyone he knew and everyone he had ever seen. It was unremarkable because it was universal in his world. The farmers who arrived from Anatolia, carrying their grain seeds and their livestock and their slightly lighter skin, did not know that their skin was lighter because of selection for vitamin D synthesis. They did not understand vitamin D. They did not understand ultraviolet radiation. They were simply people who had been farming in the sun of Anatolia for generations, and who were now moving their families and their agricultural knowledge into new territory. The Yamnaya herders sweeping across the